Good day, this is Professor Will, CBMD PhD, and today's Thursday, August 5th, 2021. It is 2:18 and 53 seconds, a.m. Eastern Standard Time. Today's research, Corona. So let's hop to it here. The study reveals what triggers lung damage during COVID-19. A longitudinal study of macrophages from SARS-CoV-2. Infected lungs offers new insights into dynamic immunological changes. Date August 4, 2021. Source: the Korea Advanced Institute of Science and Technology. Researchers found that a specific subtype of macrophages that originated from blood monocytes play a key role in the hyperinflammatory response in SARS-CoV-2 infected lungs. By performing single-cell RNA sequencing of bronchoalveolar lavage fluid cells, the study provides new insights for understanding dynamic changes in immune response to COVID-19. In the early phase of COVID-19, SARS-CoV-2 infected lung tissue, and the immediate defense system is activated. This early and fast response is called innate immunity, provided by immune cells residing in lungs. Macrophages are major cell types of the innate immune system of the lungs, and newly differentiated macrophages originating from the bloodstream also contribute to the early defenses against viruses. Researchers investigated the quantitative and qualitative evaluation of immune responses in the lungs of SARS-CoV-2 infected ferrets. And to overcome the limitations of research using patient-originated specimens, the researchers used a ferret infection model to obtain SARS-CoV-2 infected lungs, subsequently with a defined time interval. The researchers analyzed the 10 subtypes of macrophages during the 5-day course of SARS-CoV-2 infection and found that infiltrating macrophages originating from activated monocytes in the blood were key players for viral clearance as well as damaged lung tissue. Moreover, they found that the differentiation process of these inflammatory macrophages resembled the immune responses in the lung tissue of severe COVID-19 patients. Currently, the research team is conducting a follow-up study to identify the dynamic changes in immune responses during the use of immunosuppressive agents to control hyperinflammatory response called cytokine storm in patients with COVID-19. The research analysis will enhance the understanding of the early features of COVID-19 immunity and provide a scientific background for the more precise use of immunosuppressive agents targeting specific macrophage subtypes. The study is the first longitudinal study using sequentially obtained immune cells originating from SARS-CoV-2 infected lungs. The research describes the innate immune response to COVID-19 using single-cell transcriptome data and enhances our understanding of the 2 phases of inflammatory responses. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.